Hi students, welcome to Sunil's tutorial. I'm Sunil Milwani and today we'll be doing this chapter called as Control and Coordination. Now, today we are going to try to understand the ultrastructure of snaps. Let's try to see the ultrastructure of the snaps, right? Now, synapses is the psychological junction between the axon of one neuron and the dendrites of the next neuron. The axon of one neuron and the dendrites of the other neuron, the small space that is there between them is called as the synapse. So, synapses is the physiological junction. Synapse is the physiological junction between axon terminal of one neuron and dendrite of the other neuron. We've already done this in some previous lecture. What is synapse? Dendrite of next neuron, right? That is what is synapse. Now, the axon terminal of one neuron comes into close proximity with the dendrite of the other neuron, but there is no actual continuity between the two neurons. There is always a small microscopic gap, and this small microscopic gap is called as synapse. I can say that the axon terminal of one neuron comes into close proximity with the dendrite of another neuron but no actual continuity persists. There will always be a small microscopic gap. No actual continuity persists between the two neurons. Right? There will be no actual continuity between the two neurons. These two neurons are separated by a minute intercellular gap which is approximately 10 to 20 nanometers. The gap between the two neurons is going to be approximately 10 to 20 nanometers, and it is filled with a fluid, and that gap is called as the synapse. Okay, so I can say that these two neurons are separated. These two neurons are separated by a minute intercellular gap. Which is approximately 10 to 20 nanometers. Right? Now this gap is filled with fluid called the synaptic cleft. Right? Can you just see the diagram, guys? This is the gap, right, between the two neurons, and this is the fluid that is filled in between in that minute gap between the two neurons. Fine, do we get this in clear? Now, when you talk about snaps, there are two types of snaps. One is called as the chemical and the other is called as the electrical. there are two types of synapses, right? One is called as the chemical and the second is called as the electrical. These are the two types of snaps. Depending upon the nature of the transfer of information, depending upon the nature of transfer of information, depending upon the nature of transfer of information across the synapses. Fine. Now, let's see chemical synapses. Synapses. What are chemical synapses? Now, if you remember your nerve impulse, you have axon and you have dendrites, right? So you would have dendrites that would be like this, and then you would have axon that would be coming out from it, the nucleus in the center, and you would have axon that would come out from here, and then it would branch. You would have the axon branching into here, right? You would have a structure something similar to this, right? This is my axon cylinder, right? And this is where you are going to have the stimulus. Stimulus is going to be given to this, right? Now I'm going to actually see how the stimulus will pass using these nerve fibers, right? Now, the axon terminal divides into many branches. The axon terminal divides into many branches, right? Divides into many branches, and each end is a synaptic knob. So each of this end of the axon is going to be a synaptic knob. Now I will show you a magnified version of this synaptic knob. Do you understand this? This is my impulse that is going to be transmitted to the branches. Now that end of this branch, the magnified version is this. Right? This is my synaptic knob. Fine, do you get this thing clear? Do you understand what am I talking about? So the axon terminal would divide into many branches, and each branch is called as a synaptic knob. This is how your synaptic, that is going to be your tip of those branches. Now let's see how the nerve impulse moves out from there. From the dendrites, the nerve impulse must have gone into the axon, must have gone into the branches. Now I want to see from this branch, how does it go into the dendrites of the other nerve fiber? Right, that's what I'm trying to find out. The synaptic knob of each axon comes in contact. The synaptic knob of each axon. The synaptic knob of each axon comes in contact with the dendrite of the next axon. Fine. The synaptic knob contains many. The synaptic knob contains many the synaptic knob contains many synaptic vesicles. Vesicles in the cytoplasm, in its cytoplasm. And these vesicles store chemicals, and these vesicles store chemicals such as norepinephrine, and acetylcholine. Right? These are those neurotransmitter chemicals that are stored in the vesicles. Can I just show you this, guys? This is my synaptic knob. Now these synaptic knobs, here you have this is cytoplasm here. They contain many synaptic vesicles. Now these vesicles are filled with the neurotransmitter chemicals. They are filled with your acetylcholine and your norepinephrine. These are your neurotransmitter vesicles, neurotransmitter chemicals that are filled in these synaptic vesicles. So at the synaptic knob, the cytoplasm is going to have a large number of synaptic vesicles. These synaptic vesicles are filled with the neurotransmitter chemicals. Fine, do we get it till here? Now, the membrane of the synaptic knob on the axon side, the membrane of the synaptic knob on the axon side is thickened due to cytoplasmic condensation. Is thickened due to cytoplasmic condensation and is called the presynaptic membrane. Right? Here, guys, can you just see the diagram? This membrane, the synaptic knob membrane on the axon side, due to the cytoplasmic condensation, becomes a little thick, and this is called as your presynaptic membrane. So the membrane near the edge of the knob is slightly thicker than the normal membrane, and that's because of the condensation of cytoplasm, and this is called as your presynaptic membrane. Fine, do we get this much clear? Next. Now, when a wave of depolarization reaches the synaptic membrane, you have stimulus that will be given here. Guys, you would have stimulus that would be given to the dendrite that will send the impulse. Now that is going to cause depolarization, which I'm going to explain to you in a little time, right? For the timing, just let's accept that there is something called as depolarization, right? What it is, how it takes place, I'll just explain it to you a little later. So as soon as you give impulse, when the impulse is transmitted, you have a phenomena that would occur which would be called as depolarization, right? So when a wave of depolarization reaches the synaptic membrane, calcium ions diffuse into the terminal from the surrounding fluid, right? What would happen is that all the calcium ions would diffuse from the surrounding fluid into the presynaptic membrane, right? The calcium ions then stimulate the synaptic vesicles synaptic vesicles in the terminal to move towards the terminal membrane. Exactly what happens in amoeba, how a vacuole moves towards the membrane and then it gives out. So first what would happen is that the calcium ions would accumulate here. As soon as the impulse reaches the knob, the calcium ions would accumulate in the presynaptic membrane. These calcium ions would cause the synaptic vesicles to move towards the membrane, right? Once they reach the membrane, once they come in contact with the presynaptic membrane, the presynaptic membrane and the membrane of the synaptic vesicles would rupture, thereby releasing the chemical, the neurotransmitter chemical in that synaptic space. Fine, do we get this in clear? Can you just write this much down? When a wave of depolarization, when a wave of depolarization reaches the presynaptic membrane, calcium ions diffuse into the terminal from the surrounding fluid. Calcium ions diffuse into the terminal from the surrounding fluid. Right? From the surrounding fluid. The calcium ions then stimulate the synaptic vesicles. Calcium ions then stimulate the synaptic vesicle vesicles in the terminal to move towards the terminal membrane. In the terminal to move towards the terminal membrane. Fuse with it and then rupture. Fuse with the terminal membrane and then rupture vesicles for release of vesicles for the release of the neurotransmitter. Into the cleft. That's how the neurotransmitters are released into the cleft, right? These are then diffused into the synaptic gap. These are then diffused into these synaptic gaps and they combine with the specific receptor molecules of the target cells of the receiving dendrites. So this is how the impulse is going to be released. So the dendrites which is present here on the other side of the synaptic gap would receive those chemicals and would, have the nerve impulse accordingly generated. Fine, do we get this in clear? These neurotransmitters would combine only with the dendrites of the receiving organ. If there is some other dendrites, they would not combine with that. They will not have any effect on other dendrites. They would have effect only on the dendrites of the receiving organ. Fine, do we get this in clear? So you have those neurotransmitters which have been released into this gap. Next, these are then diffused into the synaptic gap. These are then diffused into the synaptic gap. These are then diffused into the synaptic gap. They then combine with the specific receptor molecules. They then combine with the specific receptor molecules. They then combine with the specific receptor molecules of the target cells of the dendrites. Of the target cells of the dendrites. Fine. Which is called as the postsynaptic membrane. Here you can, in the diagram, can you just see the diagram? In the diagram, they are combining with the receptor molecules of the target cells, and that membrane, which is in the membrane of the dendrite, is called as the postsynaptic membrane. Like how the membrane of the axon, the tip of the axon is called as the presynaptic membrane, the membrane of the dendrite is called as the postsynaptic membrane. So they combine with the target cells of the dendrite. Target cells of the dendrite, which is called as the postsynaptic membrane. Which is called as the postsynaptic membrane. Fine, do you get this thing here? Now, this combination of the neurotransmitter with the postsynaptic membrane causes sparking for a second, and the electric current due to the depolarization passes on its signal. As soon as you have the neurotransmitter combining with the postsynaptic membrane, what would happen is they cause sparking, right? That means there would be an electrical contact that would be established between them. So they cause sparking and electric current due to depolarization passing on its signal. Sparking a second and sparking a second electrical current due to depolarization passing on its signal. Right? So that's how the signal is propagated. This results in the propagation of the nerve impulse to the next neuron. This results in the propagation of nerve impulse to the next neuron. Right? To end the signal, now you already sent the signal to the next neuron. To end the signal, the synaptic bulbs reabsorb some of the neurotransmitters, and the enzymes which are present in the synapse neutralize the other. You have those neurotransmitters which are there in the synaptic gap. What do I do with them? Right? Because till the time they are there, the impulse will keep passing. So as soon as the impulse is passed, to end the signal, the synaptic bulb reabsorbs some neurotransmitters, and the enzymes in the synapse neutralize others. right? So, do we understand how chemical synapse is sent from one axon to the other dendrite? Next, what do we understand by an impulse? What is an impulse? An impulse is nothing but it is the sum total of electrochemical changes that are aroused by the stimulus. If I stimulate any of the cell, there would be certain electrochemical changes that would be aroused. The sum total of all these electrochemical changes is called as a nerve impulse. Now, because of these electrochemical changes, you would have two processes that would take place. One is called as polarization and the other is called as depolarization, right? Now, these two processes help in carrying the signal from one end to the other end in the form of a wave. So, what is an impulse? It is the sum total of electrochemical changes This is the sum total of electrochemical changes aroused by a stimulus in the form of depolarization and repolarization, which is carried along the nerve fiber to its termination and results in the propagation of wave. So basically, all the electrochemical changes that occur which enable you to send the stimulus from one end to another end is called as an impulse. Right? Now, why do we have impulse? Because excitability and conductivity is the characteristic property of the nerve fiber. Right? If the nerve fiber is like a cylinder and it is selectively permeable, why do we have impulse? Or how is impulse generated? How do I generate an impulse? I can say that excitability and conductivity. Excitability and conductivity is the characteristic property of a wave. Of a nerve. All nerve fibers have a capability of conducting impulses and also have a capability of getting excited. Now, the nerve fiber is like a cylinder with a selectively permeable membrane. The nerve fiber is like a cylinder with a selectively permeable membrane. Right? Now, guys, can you just see this? the nerve fiber is like a cylinder with this membrane. Both the membrane of the axon and the dendrite is selectively permeable. That means it will allow certain ions to pass through more than it will allow some other ions to pass through. That means there will be some ions that would pass inside more and some would remain outside. So there would be some ions that would go inside more and some would go inside less. The ones which go inside more, there will be a deficiency of them outside. The ones which go inside less, there will be excess of them outside. Fine, do we get this thing clear? That is what is selectively permeable. Now let's see which are the two ions that are responsible for this. It allows the passage of only one type of ions. It allows the passage of only one type of ions, that is, say, sodium ions or potassium ions, right? So it would allow sodium or potassium ions and conduct impulses in the form of electrochemical reaction and conduct impulses in the form of electrochemical reaction. Since it allows only one of the two ions to pass through, right? That's how that you would have a charge that would be developed, and that's how you would have an electrochemical reaction when it is stimulated. When it is stimulated chemically or electrically or mechanically, right? So that's how the impulse is going to be generated. Now let's see in detail actually how this passage of sodium and potassium ions helps in generating an impulse. First, let's see the resting phase. When there is no stimulus given to the cell, how is the nerve impulse? That is called as the resting phase. Resting phase is the non-stimulating nerve fiber of a non-stimulating nerve fiber. When there is no impulse given to a nerve fiber, the outer surface of the membrane remains positively charged. Guys, can we just see this? This is my cylinder. The outer surface is positively charged and the inner surface is negatively charged, right? The outer surface is positively charged due to the sodium ions and the inner surface is negatively charged due to potassium ions, right? It is selectively permeable. It allows more passage of potassium ions, as a result of which there are sodium ions which are present outside, that makes the outer membrane positively charged and the inner membrane negatively charged. Inner side of the membrane negatively charged. Fine, do you get this thing clear? So in the resting phase of a non-stimulating nerve fiber, has outer surface of the membrane positively charged due to sodium and inner surface negatively charged due to potassium. So in the resting phase, the outer surface is going to be positively charged and the inner surface is going to be negatively charged. The membrane at this stage is called as polarized because there is going to be some charge on it. The membrane at this stage is called polarized, and its resting potential is minus 70 millivolts. It could be somewhere between minus 40 to minus 90 millivolts, right? That would be the potential difference that would be there, that would be present on the membrane of the nerve fiber. That is the amount of that is the exact amount of potential that will be developed on the membrane of the nerve fiber. Why do I have a minus sign? The minus sign indicates that the inside is negative relative to the outside. What I write this side? The minus sign indicates that the inside is negative, relative to outside. Right? So because of selective permeability of ions, you have a potential that is developed which is nothing but which is equal to minus 70 millivolts. Right? What is actually minus 70 millivolts? It is nothing but the net difference in the charges between the inner and the outer surfaces of the nerve fiber. If you find out the difference between the inner and the outer surface, that's actually my potential difference between the inner and the outer surface of a non-conducting nerve fiber. Right? So I can say that the resting potential is the net difference in the charge between the inner and the outer surfaces. The difference in the charge between the inner and outer surfaces of a non-conducting nerve fiber. Right, that's your minus 17. Now, the membrane is highly polarized. Why does it become highly polarized? Because it has poor permeability for sodium ions and higher permeability for potassium ions. Right? The reasons why is it that you have that charge developed? First, the membrane is polarized. Polarized means it has certain charge on it. Why does it have a charge on it? The membrane is polarized due to poor permeability of sodium ions and higher permeability of potassium ions. Right, that is one of the reasons. Secondly, due to the presence of ATP. Due to the presence of ATP powered, you would have certain energy that would give an ATP sodium potassium ion exchange pump. Right? Each sodium potassium ion transport pumps because of the presence of ATP, because of the energy that is given by the ATP. What would ATP do? It would expel 3 sodium ions for every 2 potassium ions that are imported. That's what ATP does. That's the reason why, one, as it is, there is semi-permeability. There are less number of sodium ions going inside as compared to potassium. Moreover, the ATP which is inside is expelling both sodium and potassium. It is expelling sodium ions. So it is expelling 3 sodium ions and it is importing. It is expelling 3 sodium ions and it is importing 2 potassium ions. So as a result of which the concentration of potassium ion inside would increase, right? And this process is called as the sodium potassium ion exchange pump. How is this pump created? This pump is created because of the energy that is derived from ATP. Each sodium potassium transport pump expels 3 sodium ions for every 2 potassium imported. Every 2 potassium ions imported. Right? Such pumps are said to be electrogenic. So that is in the resting phase. So in the resting phase, the membrane of the nerve fiber is highly polarized. Now let's see what happens during the active phase. Active phase as in when a stimulus is given to the nerve fiber. Now during active phase, the nerve fiber is stimulated. During active phase, the nerve fiber is stimulated with threshold stimulus. What do I understand by threshold stimulus? Stimulus that requires sodium, that requires to open sodium channel. That means the stimulus should be minimum. If this is the resting potential is minus 70 millivolts, the stimulus should be at least minus 55 to minus 60 millivolts, right? And that stimulus is called as threshold stimulus. The stimulus should be capable of producing a potential of minus 55 to minus 60 millivolts. How will this potential be developed? This potential is going to be developed because of the disturbance that is going to be caused in the sodium ion concentration. How? During the active phase, the nerve fiber is stimulated with threshold stimulus. The disturbances occur at the point of the stimulus. Disturbances occur at the point of the stimulus. Disturbances occur at the point of the stimulus, and the membrane permeability is momentarily reversed. That means, guys, can you just see this? If I'm going to touch this point of the membrane, there would be a disturbance in the membrane, and as a result of this disturbance, what would happen is at that point of contact, the polarity would get reversed. I had positive charge outside and negative charge inside, here the polarity would get reversed, where I would get negative charge outside and positive charge inside. Why would this happen? Because of the disturbance that you would create in the impulse by the stimulus. Fine, do we get this thing clear? So the polarity would get momentarily reversed. The positive sodium ions and move in the nerve fiber. Positive sodium ions move in the nerve fiber. Or how is it that the polarity is changed? Because as soon as you touch the membrane, as it is semi-permeable. As soon as you touch it, its permeability is going to change. So the sodium ions would move in and the potassium ions would move out. Right? The positive sodium ions move in the nerve fibers and simultaneously the potassium ions move out. Sodium ions would move in and potassium ions would move out. So the membrane becomes negatively charged outside and positively charged inside. I can say so. The membrane becomes positively charged inside and negatively charged outside because of this movement of sodium and potassium ion. Right? In this condition, the membrane is said to be depolarized. This is your depolarized. In this condition, the membrane is said to be depolarized. Remember, we had done this word before also. I told you I'll explain this word to you later. So this is the depolarized, and the sudden change in the membrane potential is called as action potential. Obviously there is going to be a difference. The polarity will change and the potential will also change, because not all the sodium ions have gone in, not all the potassium ions have come out. So there is going to be a slight difference in the potential and the polarity. So in this condition, the membrane is said to be depolarized, and the sudden change in the membrane potential is called action potential. Right? The value of this action potential, its value is equivalent to plus 40 millivolts. The charge inside is greater, therefore plus 40 millivolts. This shift in ions and electrical charges are sufficient enough, then it results in the formation of a current sink. If this is sufficient threshold potential, then you would have something that is called as a current sink. Right? I'll just tell you what it is. Just give me a minute. If this shift of ions, that means if sufficient amount of ions are moved inside and electrical charges are sufficient enough, electrical charges are sufficient enough, then it results in the formation, forming current sink at the point of depolarization. Current sink at the point of depolarization, there would be a current sink that would be formed. Now, what is a current sink? Which you would have the membrane that would try to come back to its equilibrium position. So you would have the charge that would move down. These charges will move down, and guys, can you just see this? These charges will move down and the impulse would then get conducted. Each positive charge would get attracted towards a negative charge, right? So as a result of which the positive charge is moving ahead and the impulse gets conducted. You have this set of positive charges which are inside the oxon. Okay. Now we know that opposite charges attract, so this positive charge is going to be attracted by the negative charge. So it will move towards the negative charge. Then it will get attracted by the next negative charge, will move ahead, will get attracted by the next negative charge, will move ahead, and that's how your impulse will move from one point to another point. Fine, do you get this thing here? That's your current sink. Depolarization, which spreads a wave of excitation along the nerve fibers and is called nerve impulse. That's how your nerve impulse is generated, right? It is self-propagating wave which travels rapidly from the point of stimulation to the end of the axon. It is self-propagating wave because of the opposite charges. Self-propagating wave which travels rapidly from the point of stimulation to the end of the axon, right? So that's how you would have the wave that would move. Okay. Now the next thing that is remaining is the recovery phase, which we will do in the next lecture. Let's stop this here for the day. Thank you very much.